Soil biology is integral to soil health. Microbes living in soils can improve crops' nutritional status, reducing the need for external fertilizer inputs, and can even increase tolerance to drought, pests, and diseases. But the path to fostering high-functioning soil microbial communities is not always clear. Here at the Berkeley Agroecology Lab, we're asking the question, how can we manage soil microbial communities to improve the productivity and resilience of agricultural systems? Arbuscular mycorrhizal fungi, or simply mycorrhizae, are soil microbes that are especially relevant in agricultural contexts. They colonize plant roots and are known to provide many benefits to their host plants, such as improved access to nutrients and water. This is achieved through an extensive network of fungal filaments, providing plants with access to a larger volume of soil than they could reach with their roots alone. In exchange, mycorrhizae use plant carbon as their source of energy. In fact, mycorrhizae can't grow and reproduce without plant carbon. They are what is known as an obligate biotroph, meaning that they have to have a relationship with another organism to get their food. Mycorrhizae are ubiquitous in soils, and most plants are colonized by them by default, around 70 to 80% of all plants on Earth, including most important crops other than brassicas. There is a vast diversity of mycorrhizae that, like plants, have different growth strategies and fulfill different functions in soil ecosystems. So, just like different plants grow in different places depending on the soil, climate, and other factors, the composition of a mycorrhizal community will change depending on environmental conditions such as carbon and nutrient availability, and the level of soil disturbance. In agricultural systems, the level of disturbance is greatly affected by soil management practices. Common practices like tillage, long fallow periods, or the use of synthetic fertilizers can suppress mycorrhizal diversity and abundance. Consequently, under some types of agricultural management, the mycorrhizal community may not function as effectively as it could. Some management practices could improve mycorrhizal functioning, or even augment native mycorrhizal populations through inoculation. In fact, a variety of commercial inocula are currently available on the market, each claiming to enhance plant growth through the introduction of several strains of mycorrhizae. But will these inoculants improve mycorrhizal functioning in your field and provide the benefits like increased nutrient uptake, reduced susceptibility to water stress, and the like? Are they worth it? To answer that question, it's important to consider what mycorrhizae need to flourish. We know mycorrhizae need plant carbon to meet their energy demands. No matter how much you inoculate, mycorrhizae will still need year-round sources of plant carbon to sustain their populations. This can be achieved by growing cover crops, which provide mycorrhizae with the necessary resources between cash crops. In addition to fostering general mycorrhizal abundance in a field, it's also important to consider the diversity of the mycorrhizal species. More diverse mycorrhizal communities are more adaptable and are thought to provide greater benefits to crops. Fortunately, studies show that above and below ground diversity are linked, so mycorrhizal diversity can be built through a diverse plant community. As such, intercropping and crop rotation may foster more diverse and abundant mycorrhizal communities, which in turn will improve crop outcomes. Greater abundance of mycorrhizae can also be achieved by reducing disturbance through no-till or conservation tillage, and possibly increase diversity too. Disturbance favors certain types of mycorrhizae that are likely not the most beneficial for plants. Further, the less disturbance, the larger the mycorrhizal network can become, increasing plant benefits. And finally, mycorrhizae want what we all want, a sense of purpose. In fertile soils, plants might not be inclined to form associations with mycorrhizae, as they can easily meet their nutrient demands without mycorrhizal help. However, this will also cut plants off from the water, pest, and disease benefits mycorrhizae are also able to offer. Things can get complicated here quickly, though, and the relationship between nutrient availability and plant benefits from mycorrhizae can vary, and it's not yet fully understood. Okay, so we know what mycorrhizae need to flourish in your fields, but what about inoculating your field with mycorrhizae? What happens if there are plenty of nutrients in the soil and you inoculate? If you're already employing cover crops, crop rotations, and conservation tillage, will external inoculants interfere with the mycorrhizal community you've so carefully fostered? It's possible you already have a hardworking team of mycorrhizae in your soil. Or that the mycorrhizal species you inoculate with is a poor fit to your field conditions, or a poor match to the crop variety. In those cases, inoculating likely won't do much, but it's a challenge to predict all of those factors in advance. Hoping to get to the bottom of this, we set up an experiment on tomato fields in the summer of 2020 to investigate the effect of mycorrhizal inoculation in water-stressed tomato plants. We set up 2 experiments, one in a fresh market tomato field and the other in a processing tomato field. The fields were set up with either full or deficit irrigation, in addition to applying mycorrhizal inoculum to half of the plots. For the fresh market experiment, in both full and deficit irrigation, we found no effect of inoculation on tomato yields. In contrast to our results from the fresh market tomatoes, we did see an effect of inoculation in processing tomatoes that experienced water stress. We saw a 10% increase in tomato yields of inoculated plants under deficit irrigation compared to non-inoculated plants, while in the full irrigation treatment, inoculation actually lowered yields by 18%. Yet, other studies have found positive effects of inoculation on processing tomato yields, even under full irrigation. These varying results highlight that more research is needed to better understand the circumstances in which mycorrhizal inoculation will be effective.
In our lab, we're striving to learn more about when mycorrhizal inoculation may be beneficial. We're still analyzing data from 2020 and are getting ready for more experiments in 2021, doing trials in fields with all sorts of management histories across the Sacramento Valley and Central Coast. Stay tuned to hear more about our results. If you'd like to get involved in our research or hear more about the project, don't hesitate to get in touch with us or see what we're up to on our website or on Twitter.